Welcome back to week 9 of Applied Immunology. The first lecture for this week centered on allergic reactions, which encompass several types of detrimental adaptive immune responses that are activated in response to harmless environmental antigens, and we call these allergens. We will build on that theme with today's lecture 9b, where we will look at another set of immune pathologies resulting from undesirable activation of inflammatory responses against self-antigens. Using the Greek prefix auto, which means self, we collectively refer to these as autoimmune disorders. Today we will discuss the ways in which genetic and environmental factors promote a breakdown in immunological tolerance. We will also talk about the effector cells and tissue types that can be affected based on patterns of antigen expression, and then end by summarizing the mechanism of action of several autoimmune pathologies. Autoimmunity encompasses a wide variety of diseases that attack healthy self-tissues through several different underlying mechanisms. This table summarizes some of the most common autoimmune disorders, many of which we will learn about in depth today. These all vary with respect to the mechanism of disease pathogenesis, which, since these are mounted in an antigen-specific manner against self-tissue, these require adaptive immune activation through either autoreactive T cells or the production of autoantibodies by autoreactive B cells. For some of these, specific autoantigens have been defined, while others may be driven by a mixture of autoantigens. These diseases also vary with respect to the consequences or symptoms of immune activation, which largely depends on where autoantigen is expressed in the body. Lastly, the prevalence of each disorder varies, although it's thought that roughly 5% of the population in Western countries is affected by autoimmune disease. A recurring theme in this course is that inflammatory immune responses must be deployed against pathogens while remaining non-reactive to healthy self-tissues, which is the concept of immunological tolerance to self-antigens. We've learned that there are numerous mechanisms through which tolerance is established and maintained in lymphocytes, and that these mechanisms must be functional in order to avoid autoimmune diseases like the ones listed above. In order to learn more about how tolerance is broken in autoimmunity, let's first recap the mechanisms through which tolerance is established in the healthy patient. We can start with central tolerance in primary lymphoid organs, which refers to selection mechanisms that occur at different checkpoints during either B cell development in the bone marrow or T cell development in the thymus. These checkpoints serve to either delete or edit lymphocyte antigen receptors that bind strongly to self-antigens, or they recognize self-antigens. One important mechanism of central tolerance during T-cell development is something that we haven't talked about yet, which is the expression of a transcription factor called autoimmune regulator, or AIR. Now, selection in the thymus is based on expression of the majority of self-antigens within the thymus, but there are still some self-antigens that are only expressed in specific organs or tissues that would not otherwise be expressed in an organ like the thymus. And these are called tissue-specific antigens, or TSAs. And these include things like insulin, which is expressed in the pancreas, myelin proteins that are found in the brain, and some retinol proteins that are expressed in the retina of the eye. AIR is a transcription factor activated in thymic epithelial cells and thymic dendritic cells that actually turns on expression of these TSAs in different APC subsets. And this allows the APCs to present these antigens to developing thymocytes in order to establish central tolerance against peripherally expressed self-antigens. Central tolerance, therefore, should remove autoreactive T cell clones that recognize either thymic antigens or peripheral TSAs. If central tolerance functions perfectly, then the end result is that only non-autoreactive lymphocyte clones are allowed to develop into a mature naive state, and these cells are the only ones that are found in the pool of lymphocytes that circulate through tissues and populate the secondary lymphoid organs. There are also several mechanisms for establishing peripheral tolerance, or tolerance exerted on lymphocytes once they finish development in the primary lymphoid organs. We haven't covered all of these extensively, except for our lecture 5A on B cell development, where we learned about anergy. And again, this is a state where B cells become unresponsive to cognate antigen. There are other mechanisms of peripheral tolerance, including the physical segregation of antigen, either intracellularly, where lymphocytes can't access it, or within organs that are not surveyed by lymphocytes, which are called immune-privileged organs. And these include places like the eye, brain, and reproductive organs. Lymphocytes can also undergo activation-induced cell death, which is a form of apoptosis that occurs following repeated antigen stimulation. And this is meant to remove inflammatory activated lymphocytes from the body after they've presumably had enough time to exert effector functions to remove pathogens. Functional deviation is another thing that refers to the differentiation of T cells towards alternative or less inflammatory phenotypes, such as Th2s in some contexts or T regulatory T cells. Lastly, Tregs are extremely important in establishing and maintaining peripheral tolerance. Their immunosuppressive activities often override inflammatory signals from neighboring T cells, as the secretion of cytokines like IL-10 and TGF-beta inhibit effector T cells, including those that may be self-reactive. Tregs can also kill effector T cells, either directly through cytotoxic mechanisms or indirectly by starving them for nutrients or survival cytokines like IL-2. Tregs can also inhibit APCs through expression of regulatory ligands like CTLA4, LAG3, and IDO. Collectively, the activities of Tregs make them a potent inhibitor of potentially autoreactive inflammatory responses in the periphery, and it's worth noting that many mechanisms of Treg-mediated immune suppression are actually co-opted in the context of tumor suppression of the immune response, which we will learn more about next week in Week 10 lectures on tumor immunology. We can think of all of these mechanisms used to establish and maintain both central and peripheral tolerance as a sort of gauntlet or succession of checkpoints. And each of these checkpoints work together to prevent either the development or activation of autoreactive lymphocytes. And any individual checkpoint can catch any potentially dangerous autoreactive clones that slipped through a fault in an upstream checkpoint. And this works the majority of the time in healthy individuals. So what types of defects in these mechanisms actually occur in the patients that present with autoimmune disorders? Much like allergy, autoimmune disorders can be controlled by both genetic and environmental factors. Let's start by looking at genetic factors. So, genetic mutations that affect the presence or function of certain genes involved in immune regulation understandably influence the phenotype of certain immune cells, which could enable autoimmune disease through a number of mechanisms depending on the mutation and the tissue that it affects. These mutations may affect the survival of certain cell types or key aspects of their function that are needed to maintain tolerance, and this leads to a variety of pathologies depending again on the cell type as well as the tissues that are involved. There are a handful of rare autoimmune disorders that are driven by mutations or deletion of a single gene, and we call these monogenic disorders. These are quite rare and are often inherited in either X-linked or autosomal recessive fashion. This includes air deficiency or mutation, which again is required for tissue-specific antigen expression in the thymus. So without air, people develop a severe autoimmune disorder called APSED, which attacks peripheral organs. CTLA4 mutations are associated with Graves' disease, type 1 diabetes, and others due to decreased regulation of effector T cells. FOXP3 mutations can lead to a lack of functional Tregs, and this results in a severe autoimmune disease called IPEX, where patients die at a fairly young age. Other monogenic diseases include FAST mutations, which render T and B cells more resistant to FAST ligand-mediated killing, and this results in a lymphoproliferative disorder called ALPS. These types of monogenic disorders make sense since they involve genes that are directly required for the cells and mechanisms of central or peripheral tolerance, which we just talked about in the last slide. However, the majority of autoimmune disorders are thought to be polygenic in that they result from numerous defects in multiple immune pathways that are required for either immune tolerance or the regulation of inflammatory responses. Understanding how different gene products interact with one another in polygenic disorders is complicated, but let's look at two genetic factors that are often implicated in autoimmunity. The first of these is quite straightforward since it is biological sex. This graph shows incidence of diabetes in a mouse model of type 1 diabetes called NOD, where you can see that female mice in red have earlier onset and higher penetrance of type 1 diabetes compared to male mice of the exact same genotype in the blue line. So again, these are genetically identical individuals except for their difference in sex. This is obviously an example from a preclinical animal model used in research, but it's worth noting that autoimmune disorders in general tend to occur at higher rates in women than men. This is an example of sexual dimorphism. This is particularly true for systemic lupus erythematosus, or SLE, as well as multiple sclerosis, or MS. The mechanisms underlying why this happens are still unclear, but sex-dependent mechanisms of autoimmunity are a major area of ongoing research. HLA haplotypes are another genetic factor that is associated with susceptibility with some autoimmune disorders. This table lists MHC alleles that are strongly associated with particular autoimmune diseases, presumably because these MHC alleles exhibit especially strong binding to an epitope of self-antigen that's involved in that autoimmune disease. Alternatively, certain MHC alleles may influence the TCR repertoire during establishment of central tolerance in a way that sort of allows for maturation of more autoreactive T cell clones, as would be the case if MHC bound weakly to some self-antigens expressed in the thymus and therefore didn't exert sufficient negative selection on thymocytes. The exact mechanism underlying these types of associations likely depends on the specific disease context, but nevertheless, HLA haplotype is a recurring genetic locus that is associated with susceptibility towards autoimmune pathologies. The second type of factor implicated in autoimmune disease development are environmental stimuli, such as infections or injuries that induce an inflammatory state in the body. There are interesting correlations in patients where a bad inflammatory event preceded the onset of autoimmune pathology, and there are a few different proposed mechanisms for this. The first is that self-antigens presented during homeostasis will not stimulate T cell activation because these only consist of signal 1 of TCR activation without any co-stimulatory marker expression or cytokines that are needed to fully activate T cells. However, self-antigen that's presented by some APCs that are also activated by a concurrent inflammatory reaction could mistakenly lead to the activation of autoreactive T cells, which are unable to differentiate between inflammatory cues that are associated with pathogen-derived antigens versus self-antigen that is presented in a sort of coincidentally inflammatory context. In individuals with successful immune tolerance mechanisms, this would be a rare event because it would involve the encounter with one of only a few autoreactive T or B cell clones, but in a genetically susceptible individual, this may occur more frequently. The second mechanism has to do with infection or injury that induces tissue damage, which releases self-antigens that haven't been used to establish immune tolerance. This could involve antigens that are either sequestered intracellularly, and so they're not usually available to lymphocytes, or entire organs containing antigens that aren't usually surveyed by lymphocytes. And the latter example involves what we call immune-privileged sites that aren't typically surveyed by lymphocytes, at least not to the same extent as other tissues. And these include organs like the brain, eye, ovaries, and testes. An example of this type of injury is something called sympathetic ophthalmia, where injury to one eye leads to retinal antigen drainage to the lymph node, and this primes T cell responses that can then traffic to the other uninjured eye to attack antigen-expressing cells over there. Lastly, there are some pathogens whose antigens closely resemble certain self-antigens. This is a case called molecular mimicry, where the immune system is mounting a response to foreign antigen, but the T cell receptor or B cell receptor just happens to be cross-reactive with self-antigen. An example of this is rheumatic fever, which can occur following infection with streptococcus bacteria. In this case, an epitope in the streptococcal bacterial cell wall is structurally similar to antigens that are expressed in the valves of the heart. So these anti-streptococcus antibodies cross-react with the heart valves and cause inflammatory tissue damage in the heart. There's no actual breakdown in tolerance or immune recognition here. It's just an unfortunate coincidence that antibacterial antibodies happen to cross-react with self-antigen. The exact mechanism of autoimmune initiation depends on the specific patient and the specific autoimmune disease. So as scientists and clinicians, we try to attribute a disease etiology to a clearly defined root cause or root causes, but this just may not be possible for most types of autoimmune disease. In most patients, autoimmunity is likely caused by some combination of both predisposing genetic factors as well as inflammatory environmental triggers like infection or injury. Regardless of the underlying causes, all autoimmune pathologies are characterized by some degree of immune dysregulation that leads to the pathological activation of immune responses against self-antigens. Before we get into the specific mechanisms of action of several autoimmune disorders, I want to touch on the fact that these pathologies often involve multiple components of an immune response. This table summarizes how autoreactive T cells, B cells, and secreted antibodies all play a role in the pathogenesis of common autoimmune disorders like SLE, type 1 diabetes, myasthenia gravis, and multiple sclerosis, each of which we will discuss later on in today's talk. Although the specific underlying mechanisms may vary, most autoimmune diseases involve a breakdown of immunological tolerance of both T and B cells. For B cells, this is characterized by presentation of self-antigens to T cells and/or the generation of autoantibodies. For T cells, this can be mediated through direct cytotoxic effects of Th1s or cytotoxic T lymphocytes or CD8 T cells, as well as T cell help to B cells to promote class switch recombination and tailoring of autoantibody responses. So, although we tend to categorize some autoimmune disorders due to the primary effector functions that drive autoimmunity, keep in mind that for most of these diseases, autoimmunity is quite comprehensive in that it involves a loss of tolerance by multiple lymphocyte populations. With that being said, most autoimmune disorders can be grouped according to the predominant mechanism that drives autoimmunity. This chart organizes several autoimmune diseases according to whether the pathology is primarily exerted by autoantibodies that are bound to surface antigens, antibody-antigen complexes that form aggregates responsible for driving inflammation, or T-cell-mediated autoimmune diseases. Note that for some disorders, like rheumatoid arthritis, multiple immune mechanisms are implicated in parallel, which is why it is categorized as both an immune complex and T-cell-mediated disease. The consequence or pathological characteristics of each disorder is described in the far right column of this chart. Much like allergy, autoimmune diseases present with a wide variety of clinical symptoms that depend on the tissues where autoimmune responses are activated. It's also interesting to note that many autoimmune disorders usually present as cycles of flare-ups called relapses that are separated by periods of reduced disease symptoms called remissions. And these are called relapsing, remitting courses of disease that may continue in this cycle for months or years or even decades. Although both the stimuli that induce symptom relapse and the regulatory mechanisms that induce symptom remission, even when autoantigen is still present, both of these are largely unknown. In many cases, these diseases are treated with anti-inflammatory medications such as monoclonal antibodies against TNF-alpha or integrins involved in lymphocyte trafficking, although these immunosuppressive therapies understandably lead to off-target effects that increase patient susceptibility to certain types of infections. Since classical autoimmune disorders occur in an antigen-specific manner because they're mediated by lymphocytes, the organs where autoimmune pathology is concentrated unsurprisingly depends on where autoantigen is expressed. Some diseases are driven by a tissue-specific antigen that is not expressed widely throughout the body, so not surprisingly, autoimmune pathology is limited to the tissue or organ where that autoantigen is expressed. Examples of these would be type 1 diabetes, which specifically targets antigens expressed by beta cells in the pancreas. Another example of this would be Hashimoto's disease, which attacks the thyroid due to self-reactivity against thyroid peroxidase, which is an enzyme that's only expressed in the thyroid. Other autoimmune diseases are spread across tissues throughout the body, and so they are referred to as systemic autoimmune diseases. Antigens that drive systemic pathologies are usually things that are more ubiquitously expressed by many cell types. In SLE patients, this includes autoantibodies against chromatin or other nuclear proteins. And since these are expressed in many tissues, SLE pathology affects diverse organ systems, including the skin as well as the kidneys. For the remainder of this lecture, we will drill down into understanding a bit more about some of these individual autoimmune pathologies one by one. The first example we will look at is something called myasthenia gravis, which is an autoimmune disorder that affects peripheral nerve signals that act on muscles. This disease involves both autoreactive T cells and B cells, though the primary pathological effector are the B cells, which produce antibodies specific for acetylcholine receptor, and this is expressed on muscles. The T cells are primarily CD4s that provide T cell help to the acetylcholine specific B cell clones. The autoantibodies function by binding to acetylcholine receptors on muscle cells. And this leads to the internalization and degradation of the receptor. Muscle cells therefore cannot respond to the neurotransmitter acetylcholine, which is released by peripheral nerves that innervate skeletal muscle. And acetylcholine is a key signal involved in inducing sodium ion influx to mediate muscle contraction. Patients with this autoimmune disease exhibit muscle weakness that gets progressively worse and can be potentially fatal. Myasthenia gravis is a disorder that is characterized by autoantibodies that bind to a cell surface antigen, in this case a receptor expressed in muscle tissue, and this blocks its function and leads to disease symptoms specifically within skeletal muscle. Another disorder that's primarily driven by autoantibodies is systemic lupus erythematosus, or SLE. SLE is caused by autoantibodies that recognize broadly expressed antigens. These are oftentimes nuclear antigens such as self-nucleic acids, chromatin, or other nuclear proteins. These become exposed when dying cells aren't cleared properly by phagocytes and are allowed to sort of fall apart or dissociate in tissues and release their contents. And this kind of demonstrates how important the tolerogenic signals associated with apoptotic cell clearance are for suppressing inflammatory autoimmune responses. This diagram considers SLE where the autoantibodies recognize self-RNA and self-DNA. In this case, antibodies are bound to a certain type of dendritic cell called a plasmacytoid DC, or PDC. And these are particularly good at mediating antiviral responses since they produce a lot of type 1 interferon. In SLE, however, PDCs can phagocytose antibody-antigen complexes through Fc receptor binding. And once these complexes are transported into endosomes, the nucleic acids that are bound to the antibody can activate endosomal TLRs that recognize nucleic acids, including TLR7 and TLR9. TLR activation by these cells triggers type 1 interferon production that can then act on other cells. Note that this entire process is abnormal since in healthy individuals, dying cells are cleared quickly enough to not be found extracellularly, and these ligands don't usually find their way into PDCs without the presence of anti-nucleic acid autoantibodies. Type 1 interferon produced by PDCs can then stimulate neighboring myeloid cells to produce BAFF, which is a survival ligand for B cells. In this way, type 1 interferon indirectly supports the survival of autoreactive plasma cells that are located in inflamed tissue, and this results in a feed-forward loop of autoantibody production. Because the nuclear antigens in SLE are ubiquitously expressed, they form many small immune complexes that are not cleared by phagocytes. Instead, these immune complexes circulate throughout the blood, and they get deposited in small capillary walls, joints, and this is really important, but they also tend to stick in the glomeruli of kidneys. And once they're located here, these immune complexes interfere with renal function and induce inflammation. This tissue section from a SLE patient shows an affected glomerulus where immune complexes have triggered the thickening of the basement membrane, and this is shown as the thickened sort of clear areas surrounding the glomerulus at the center of the slide. SLE patients therefore show systemic symptoms including skin inflammation, but the impaired function of the kidneys is a serious concern that can lead to renal failure in these patients. The next few autoimmune disorders that we will look at are those driven by pathologically activated T cells. This includes type 1 diabetes mellitus, which is caused by autoreactive CD8 T cells whose TCR recognize autoantigens that are expressed by beta cells, and beta cells are found in the pancreatic islets of Langeron. Beta cells produce insulin, so when CTLs recognize and kill beta cells, the pancreas no longer makes insulin, and this is what leads to the symptoms of diabetes. In mouse models of type 1 diabetes, insulin itself can serve as the autoantigen that's recognized by CTLs, although other beta cell antigens may be recognized in humans as well. The destruction of beta cells is obvious in these tissue sections on the bottom here, which compare a healthy mouse pancreas on the left to a diabetic mouse pancreas on the right. The brown cells are stained using an antibody against insulin, which marks beta cells, and these are almost entirely eliminated in the diabetic mouse. It's hard to see in this figure, but the diabetic pancreas on the right also has more T cell infiltration into the islet, and these are presumably effector CD8s that are responsible for wiping out all of the beta cells. Type 1 diabetes is distinct from type 2 diabetes, which is instead acquired in response to dietary changes and is not an autoimmune disorder, so try not to get these two mixed up. Type 1 diabetes is a good example of an autoimmune disorder where autoreactive CD8s are directly responsible for killing off healthy cells. T cells can also act indirectly by activating other immune cells to mediate autoimmune symptoms. And this is the case in rheumatoid arthritis, or RA. RA pathogenesis begins in the synovium, which is a type of connective tissue that lines the joint capsule around bone joints. It's unknown what events trigger inflammation at the synovial membranes, but once inflammation is established, it allows for circulating leukocytes, including T cells, to enter into the synovium. Once they're there, autoreactive Th1 and Th17 CD4 T cells can activate macrophages, and this in turn leads to macrophage production of pro-inflammatory cytokines like IL-1, TNF-alpha, and IL-6. These cytokines work by activating synovial fibroblasts, which then produces an important signal called rank ligand, or rankle. Now, rank ligand is significant in RA because it stimulates the differentiation and activation of mature osteoclasts. And these are a type of bone-resident macrophage that functions by eating away at cartilage and bone in the affected joint. This leads to destruction of the joint, accompanied by lots of inflammation, and this is really painful and decreases joint mobility. RA therefore leads to chronic pain, loss of joint function, and eventual disability if bone decay progresses severely. RA is a little bit complicated because numerous cell types and signaling molecules have been implicated in its pathogenesis. This simplified model shows a connection between CD4 T cells, macrophages, fibroblasts, and osteoclasts, where the T cells function in an indirect manner by activating other effector cell types. B cells have also been implicated in the development of RA, and although the specific autoantigen involved hasn't been identified yet, autoreactive antibodies and immune complex formation may be important for triggering the synovial membrane inflammation that's responsible for initiating RA pathogenesis in the first place. The last autoimmune disorder that we will discuss today is multiple sclerosis, or MS. And this is a progressive neurodegenerative disorder that affects the brain and spinal cord of the central nervous system, which we abbreviate as the CNS. MS is also a T-cell mediated disease that is largely driven by Th1 and Th17 CD4 T cells, and these are specific for various antigens expressed in myelin. And if you remember to some other physiology or maybe even neuro courses, myelin is the fatty protein sheath that covers over neuron axons and is important for action potentials. Like rheumatoid arthritis, multiple sclerosis is initiated by an unknown inflammatory trigger. Although in MS, this inflammation targets the blood-brain barrier, which is the sort of endothelial lining between circulation and the blood tissue that usually is responsible for excluding circulating leukocytes from the CNS. And this is why we refer to the brain as being an immune-privileged site. Upon opening of the blood-brain barrier by inflammation, myelin-specific CD4s can enter the CNS. And once they're here, they re-encounter myelin epitopes that are presented by brain-resident macrophages called microglia that express MHC class II. This leads to the production of inflammatory cytokines like interferon gamma from the T cells and other cytokines from microglia. And this generates more inflammation that recruits even more leukocytes. Soluble factors such as complement proteins and circulating antibodies can also infiltrate the brain, and although this includes myelin-specific antibodies, their role in disease pathogenesis is still unclear. MS involves the activation and recruitment of many different cell types into the brain, but it's unknown exactly which of these effectors are directly responsible for targeting neuronal myelin. It's thought that the combined inflammatory functions of all of these effector cells and molecules somehow leads to the removal of myelin from neuronal axons, and this is a process called demyelination. This leaves the axon vulnerable to destruction, and the resulting neuronal loss is characterized by the formation of hard or sclerotic lesions that develop in the central nervous system of MS patients. Because this is a neurodegenerative condition, as neurons are a non-proliferating cell type, patients exhibit a variety of neurological symptoms like muscle weakness, blindness, and even paralysis. Like with many autoimmune pathologies, MS patients typically start off with years and even decades of relapsing, remitting disease course, although most will eventually enter a second state of steady neurologic decline later in life, and this second phase is called secondary progressive MS. To summarize today's lecture, we've learned that autoimmunity encompasses a variety of diseases that all result from the pathological activation of inflammatory immune responses against self-antigens, which are expressed by healthy tissues, and this can lead to some pretty devastating clinical symptoms. We started off by recapping mechanisms of central and peripheral tolerance and immunity, which work in the majority of patients to prevent activation of autoreactive T and B cells. However, in autoimmune disorders, these mechanisms of tolerance are either prevented or broken through a likely combination of genetic and environmental factors. We also established some broad categories of different types of autoimmune disorders based on the predominant effector cell types, which are either autoreactive B cells or autoreactive T cells. We also subdivided autoimmune disorders based on those that affect specific organs due to limited antigen expression, in comparison to those that act systemically throughout the body by targeting more ubiquitously expressed self-antigens. We then ended today by going over several examples of the most common types of autoimmune diseases and discussed their underlying immune mechanisms and how they lead to the specific symptoms associated with each autoimmune disorder. In many cases, these types of pathologies lead to pretty severe clinical symptoms, highlighting the need to understand mechanisms of autoimmunity in order to design more effective therapies to help cure disease. If you're interested in learning more about the types of medications that are used to treat specific autoimmune diseases, I recommend that you respond to this week's discussion board prompt as one of your graded discussions for this course. That's it for today's lecture. Here's a gentle reminder that you do have one remaining lecture in week 9, which will cover the topic of transplantation immunology.